started because we still have an exciting fourth session for the day, and I'm glad to see that a lot of people are still having the stamina to come here in person and not just sit in front of their screens. So in this session, we will look at the BGI Stomics technology, and this is kind of tying everything together that we heard about today. So we talked about the high resolution but probe-driven in situ technologies. We heard about the whole transcriptome, unbiased but lower resolution technologies, and Stomics is really trying to marry these two aspects together. But we also have a panel discussion after that. And just a little housekeeping note for all the speakers that presented today: if you are around the building or nearby, we do want to present some certificates at the end of the panel discussion. So if you are able to make your way here, that would be really nice. So let's get started. I want to introduce the two speakers for the first part of the session. The first speaker will be Dr. Jiyoti Verma from Decode Science, and she is a field application scientist at Decode. And our second speaker is Dr. Dane Vassiliadis, and he is a postdoctoral fellow in the laboratory of Mark Dawson at the Peter MacCallum Cancer Center. Dane studied the role of epigenetic factors in the control of gene expression and phenotypic outcomes following environmental challenges. And recently, he has developed cutting-edge cellular barcoding methodologies to investigate the role that epigenetic priming and adaptive processes play on the initiation, the maintenance, and evolution of cancers such as acute myeloid leukemia, particularly in the response to therapy. And with that, I'll hand over to Jiyoti.
Thank you. Good afternoon. I'm very excited to be here and present overview of BGI spatial profiling technology, branded as Stomics. So last week in Cell, the spatial profiling of mouse embryogenesis was published using BGI spatial technology, and I would highly encourage you to have a look at it if you haven't done that. So before I provide the Stomics overview, very quickly I'll show you the range of our single cell and spatial NGS products that can support entire workflow from library prep to data analysis. This includes Parse Biosciences single-cell RNA-seq kits, BGI spatial transcriptomics kits, and we offer MGI sequencing platform with high-throughput sequencer and single-cell and spatial data analysis tool from Parse Biosciences and BGI. So the BGI spatial enhanced resolution omics sequencing, in short called StereoSeq, is branded as Stomics. This is an in situ mRNA capture technology for whole transcriptome profiling, which is based on DNA nanoball and provides very high resolution and large field of view. It supports simultaneous spatial transcriptome analysis of your tissue sample at a single cell, subcellular, and molecular level. The Stomics chip design uses DNA nanoball technology, which is similar to MGI sequencing platform. So what we do on a modified silicon surface, DNA nanoballs are deposited containing the spatial coordinate information with poly-T capture oligos. This allows unbiased in situ capture of all mRNA from a tissue section. The space between DNB capture spot is 500 nanometer from center to center, and the spot size is about 220 nanometer in diameter. These distances provide up to 400 spots per 10 micron square for tissue mRNA capture, which is an average size of a mammalian cell. And a 1 centimeter square chip has up to 320 million spatially barcoded spots. So StereoMix chips come in variety of sizes, and we can customize them for different tissues, such as large tissue section from monkey and human organs. In this way, we can carry out whole tissue spatial profiling that has never been studied before. The 1 centimeter square chip is already available right now, which is big enough for most users. So this is how the StereoSeq workflow looks like. It involves tissue collection and embedding, tissue sectioning and mounting on the chip, and then you perform StereoSeq experiment, which involves library prep, sequencing, and data analysis as well. So we have performed the StereoSeq on several fresh frozen tissue of mouse as listed here, and other animals including human tissue, zebrafish, amphibia, and plants. In addition, StereoSeq can enable spatial profiling of large organ and microstructure. For example, shown here, you can see the epidermis, dermis on developing limb during mouse embryogenesis, which requires high resolution. And important to note here is that the high density of StereoSeq capture spots allows spatial profiling of cell microenvironment, cell migration, and cell developmental trajectory. So this slide shows the BGI StereoSeq capture rate in comparison to the published data of other spatial technologies using tissues like mouse olfactory bulb, embryo, and liver. So the bar charts represent UMI in specific size captured by existing spatial profiling technology. So orange bar here in these graphs represent StereoSeq UMIs. So since different technologies have different capture area, we compared according to their capacity. So as shown here, the StereoSeq can capture at every resolution the highest number of UMIs. For example, in the middle, if you look at the middle chart, in a 10 micron diameter area, median 550 UMI were captured by SlideSeq version 2 using mouse embryo section, and 848 UMIs were captured by SeqScope. And as comparison, we could capture 1450 UMIs by BGI StereoSeq technology. And this is another example which shows cellular resolution in adult mouse brain can be achieved by BGI technology. Here the red dot shows the detected mRNA by StereoSeq, and blue dots are nucleic acid dye-stained mRNAs. So each blue dot represents a single cell here. So as you can see, the blue and red dots are highly overlapped, suggesting that StereoSeq captures transcript at cellular resolution. In addition, BGI have released several bioinformatics tools to support the analysis of StereoSeq data. Stereo analysis workflow is available on GitHub, which can support analysis from the beginning when you get this sequencing result. And it provides tools for mapping of the sequencing data with coordinate information on the chip. Then you can do tissue register, tissue cut, and saturation analysis, and so on. Another online tool is called StereoMap, which allows you to visualize the data by selecting the whole tissue section or specific region of your interest, and allows transcriptome clustering and statistics. So the images, as you see in this slide, are generated using the StereoMap tools. And this slide shows the list of preprints and research articles already published using StereoSeq technology, which includes studies on mouse embryogenesis published last week in Cell. As you can see here, StereoSeq technology has been used to study Drosophila embryo and larva development, solid liver tumor, zebrafish embryogenesis, and the molecular complexity and the regeneration ability of axolotl brain upon an injury. And the last one paper shows the structure of Arabidopsis leaf. And these are the things which are in R&D stage currently. For now, we can only use fresh frozen tissue, but the version 2 stereo-seq technology would be compatible with PFA, HNE, immunofluorescence, and FFPE processing. And you will get automated stereo-seq library system and a new RT reaction and library prep optimized for faster and easier spatial capture in more tissue types. Analysis tool will also include higher efficiency for big data, cell segmentation, algorithm, and total solution for 3D reconstruction. And with that, I will finish now. Thank you so much, and I'll give it to Dane.